rescue therapy, why we call the leucovorin rescue therapy. As we wait for a few more to join us, why is leucovorin rescue therapy? Why do we call leucovorin rescue therapy quickly? Quickly, class, leucovorin, we were looking at the antifolates, antimetabolites, and we started with the antifolates. And we look at antifolates, we did mention that the folic acid pathway helps us to synthesize the cofactors. Tetrahydrofolate is used as a carrier, as a one-carbon carrier moiety for the synthesis, of course, of purines, for the synthesis of pyrimidines, like thymine. And once you synthesize the purine, the pyrimidines, and of course the purines, then you can be able to synthesize the DNA. The minute, therefore, you do not supply these cofactors, there will not be purine or pyrimidine synthesis, and therefore you will not have DNA synthesis, leading, of course, to the death. So I had the last slide is that the last point we said is that you can give leucovorin, and we called it rescue therapy. And I'm asking, why do we call it rescue therapy? I asked you to find out. Did you find out? I can't see anybody responding. Why is leucovorin rescue therapy when you're using methotrexate? Why is leucovorin methotrexate? I'd ask you to look it up. Can you hear me, students? All, can you hear me? Nobody is responding. Can you hear me? Or you are looking up the answer? So let me move on as you need to be answering these questions within a second or a minute so that we proceed. And I had asked that question like last time, not now. I asked you to find out why we actually leucovorin to kind of alleviate or reduce the toxicity of methotrexate. I was hoping that you can answer it very quickly. Okay, so I'll move on because you don't seem to be responding, and so you will check for yourself why leucovorin is used as rescue therapy. And once you find out, and if it's next time, then I will discuss it. So let's move on the. We are looking at the anti-cancer drugs. I gave you a question on the alkylating agents, so we started on the antimetabolites. And we did mention the antimetabolites are drugs that have structural resemblance with some endogenous metabolites. So therefore, they can be taken up by the specific enzymes. And once they are taken up by specific enzymes, because they actually do not look like the endogenous precursors, they will stop the process. They will enzyme. Or sometimes after they are phosphorylated, they can be taken up. Once they are taken up into the synthesis of the DNA, they are taken up into the DNA because they will lack the point for continuity, they will terminate the synthesis of the DNA. So that's where we are looking at antimetabolites. We were looking at antifolates. And when we were looking at antifolates, we did mention there is that enzyme, which is very important, the thymidylate synthetase, whereby, remember, you have uracil. Uracil must be converted to thymine. And to be converted to thymine, you require that enzyme, thymidylate synthetase. Therefore, if you have drugs that can block, antimetabolites that can block that enzyme, you can therefore then stop the synthesis. And therefore, we are saying inhibitors of the thymidylate synthetase, and we have methotrexate as an indirect inhibitor. This is because when you give methotrexate, remember, it will stop the synthesis. It is an antifolate. It will stop the synthesis. It will block the dihydrofolate reductase enzyme, stop the synthesis of the cofactors. Once you have minimal cofactors, which you require to carry the methyl group to synthesize thymidine, then therefore it is an indirect, that's why you're calling it an indirect, it has an indirect effect on the thymidylate synthesis because it will lower the cofactors, and you require those cofactors if thymidylate is going to work effectively. So it is an indirect method of blockade. Then we have another drug, the 5-fluorouracil, and this one directly inhibits the enzyme thymidylate synthetase, is converted. But remember, most of these drugs as they are usually prodrugs, because they normally act when they are changed or converted into the nucleotides. They require to be converted into the nucleotides for them to have activity. So it's fluorinated analog of 2'-deoxyuridylic acid monophosphate. So you see, so it is converted. It is converted in the body to the fluorinated analog of 2'-deoxy. Remember the deoxy. DNA or DNA, which is a deoxynucleotide, it does not have the hydroxyl group at position 2'. And we are saying it must be phosphorylated. So first, the deoxy, but of course it's fluoro, because at this position here, which is position number 2, it does not have a hydrogen, it has a fluoro. So that's why we are saying fluoro. And then for the deoxy, it is because at position number 2, if I were to use just this, at this position number 2 here, it will lose this hydrogen, and there'll be hydrogen, and that's why we are calling it deoxyuridine monophosphate, which then comes with the enzyme and the cofactor terminating DNA synthesis. So in other words, this complex, that complex will block the thymidylate synthetase. Once it blocks that, the enzyme, of course, will not function, and you will not get thymine. You do require thymine for the synthesis of DNA, and that will stop the process. So also we have another drug, capecitabine, but this is a prodrug of 5-fluorouracil. So we are looking at this, and of course, if this is a hydrogen, we have uracil. If that is fluorine, we have 5-fluorouracil. And all we are saying is that drug is converted into the monophosphate. And for it to be converted into the monophosphate, at this position here, you must have the deoxy, it is monophosphorylated. So that is the deoxy UMP. And then this is the one which blocks the thymidine synthetase, and so this is the one which is acted on by the thymidine synthetase. And once, sorry. That, so if, of course, if this is a hydrogen, need to be acted on by the thymidylate synthetase to give us deoxy TMP. But of course, if this is a fluoro, it will give us the fluoro deoxy UMP, which is an inhibitor. This is the other drug that we have talked about, and we have said capecitabine is a prodrug. You can see it has structurally the same. It has the same structure of the base here. The pyrimidine is fluorinated. There is a cleavage there. And you get your 5-FU, and again the 5-FU, of course, brings about the same blockade. So this is just a prodrug which can be converted to a 5-FU. I believe we'll be able to look at that. So we are saying that the pyrimidine derivative 5-fluorouracil blocks the conversion of uridine to thymidine. Remember, where we are calling them uridine, thymidine, they must be in the form of nucleotides. For the activity to occur. The normal biosynthesis of thymidine involves methylation of the 5 position. The replacement of the 5 position, so the replacement of the hydrogen at 5 position with fluorine results in the antimetabolite. So again, I'm only further stressing on that. And of course, that antimetabolite will block our enzyme, thymidylate synthetase, and of course, block the process. Drugs targeting this enzyme selectively inhibit the formation of DNA, because thymidine is not a normal component of RNA. Remember, RNA has uracil, not thymidine, so it uses uracil, not thymine. Thymidylate synthetase is responsible for the ductive methylation of deoxyuridine monophosphate by 5,10-methylene tetrahydrofolate to yield the deoxy TMP and dihydrofolate. Remember the process require that enzyme. It is the enzyme responsible for converting UMP to the T, uridine to thymine. That's the only difference we have there. That's all you're saying. You have your 5-fluorouracil. It is converted to the nucleotide. That's here. They've only added a sugar there, so that's a nucleotide. And then it is phosphorated to the triphosphate. And once it is phosphorated, it can be taken in the RNA. Then again, it can be converted because remember here, there, it has a hydroxyl group. Hydroxyl group makes it ribose. It can also be converted, again, phosphorylated, and this position using ribonucleotide reductase converted to the deoxy, again, at a phosphorylation, and of course that will affect or will block the synthesis of DNA. The main mechanism of 5-FU activation is conversion to the fluorouridine monophosphate, and we have already mentioned that. And that can either happen, here I only want to mention the enzymes responsible. So conversion, so the conversion of 5-FU to 5 directly by orotate phosphoribosyl transferase with phosphoribosyl pyrophosphate being the cofactor. That's one way 5-FU can be converted to 5-UMP, or indirectly via fluorouridine through the sequential action of uridine phosphorylase and uridine. So these are the different ways in which the 5-FU can be converted to the UMP, or because it must be phosphorylated, or to the fluoro-UMP, it has to be phosphorylated before it activity. And this is just simply, I'll send you this, just showing the various ways in which you can start with your 5-fluorouracil. It's my 5-fluorouracil here. Here it is. We start with 5-fluorouracil, and we are saying you must convert it to the 5-fluorouracil monophosphate, and then you have the uridine kinase, which phosphorylates. So this is the UMP. You require the uridine kinase. You to phosphorate further. This is monophosphate, diphosphate, and then you require the ribonucleotide reductase now to convert the ribose sugar to the deoxyribose sugar. So this is exactly just what we were mentioning. And of course, we are saying this will be the compound or the anti-metabolite which will deactivate or which will inhibit thymidine synthetase. And once it inhibits thymidine synthetase, it will inhibit the formation of this very important cofactor, and that important it will inhibit when it inhibits thymidine synthetase. It will inhibit the synthesis of the deoxythymidine monophosphate. Once you do not form the deoxythymidine monophosphate, then for sure you will not synthesize your DNA. And once you do not synthesize your DNA, of course, the cell will die. Cannot replicate, it cannot grow, it cannot replicate. So I'll send you that as just a summary. This is just a summary of the influence of the 5-FU. It's a prodrug, must be activated into the active anti-metabolite, which can either be the triphosphate or it can be the monophosphate. And both the triphosphates, remember the triphosphates are taken in by the DNA. The monophosphate of the enzyme. So please remember those two different ways of blocking the DNA synthesis. Then we have the prodrugs of 5-FU, and of course the prodrugs, it means that these are drugs that can be acted in. You take them as being inactive. You take them as being inactive, then they are taken through position. 2' and then of course phosphorylation. So 5-FU requires intravenous administration. Of course, one of the most expensive routes of administration, and also very dangerous, and because of danger of infection and so on, is the intravenous administration. It's also very expensive in terms that the patient must go to hospital, they must have trained personnel, and I believe you have done all those things. So once that you cannot, you must administer IV. You try to see whether you can do structural modification to get other compounds that you can administer in the cheapest way, which is the oral administration. So oral products that circumvent this limitation include, we have Tegafar, capecitabine, and doxyfluoridine. So we're going to look at all of them. The first one is Tegafar. It's completely absorbed in the GIT and metabolized to 5-FU through two major pathways. The first one is where you undergo hydroxylation of carbon 5 prime, hydroxylation of carbon 5 prime of the sugar moiety, and then spontaneous decomposition. So you get hydroxylation of carbon 5 prime position of the tetrahydrofuran. This is the tetrahydrofuran moiety, and then followed by spontaneous decomposition to give us 5-FU and succinic and aldehyde. So we are saying the reason for giving the prodrug is to give us oral administration. You can also get hydrolysis catalyzed by cytosolic hydrolysis, that is pyrimidine nucleotide phosphorylase, giving 5-FU and 2-tetrahydrofuryl phosphate. So I believe I have shown those ways. So you can see the first one, these are Tegafar. So you can see Tegafar here. This is what we are calling tetrahydrofuran. We are calling it tetrahydrofuran because you can see, yes, it has an oxygen. If it was furan, it would have a double bond here and a double, because it is saturated furan. Then we are only saying it can undergo oxidative hydroxylation. This is catalyzed by the cytochrome P450 enzymes. So the tetrahydrofuran is the one which is undergoing hydroxylation, and then after the hydroxylation, this wavy line here means it can either be can carry either the alpha or beta orientation through electron shift, and then this comes in there. So when this comes in here, this now becomes a double bond. This cannot stay, so that must move in there. When that moves in there, this carbon.
Hello, Dr.
Yes.
We can't see the cursor.
Okay. Wow, sorry then. Let me get it just now. Times I forget. Thank you. Just a minute. There was somewhere I was making it bigger. Okay. So what I'm saying is this is our tetrahydrofuran. It is our tetrahydrofuran because of the fact that the furan has a double bond there and a double bond there. So there is saturation of the furan. So therefore, tetrahydro, because you have added 4 hydrogens. So now that the first way you can convert Tegafar to 5-F, the oxidase, this is the enzyme. This P450 stands for cytochrome P450 enzymes. They bring about oxidative hydroxylation. So this is where we are, of course, saying our oxidative hydroxylation. And after the oxidative hydroxylation, there is spontaneous degradation. And I'm saying the spontaneous degradation is occurring through electron movement. So you can see this here moving there. You can see that very clearly. If that moves there, this carbon, you get a double bond here. This carbon will have 5 bonds. That can't happen. So there is a cleavage of that bond. So that bond will cleave. The electrons will move on to here. If that happens, it will mean that this carbon will have 5 bonds. That can't happen. So that again breaks. I hope you have followed. This moves here. You get double bond. You can see that double bond there. Right, class? Then once this will get double bond here, it means this carbon here will have 5. So you must break this bond. And in breaking that bond means the covalent electrons are free. They move in here. Once they move in here, it means again this carbon will have 5 bonds. That can't happen. You have that cleavage there. So again, you can see this oxygen here also has a double bond, and it is that oxygen there. So you have double bond here, CH2, double bond there, CH2, carbon, double bond, cleave. And of course, you have now your 5-fluorouracil. I hope that is clear. So it's a spontaneous. It does, when we say it's spontaneous, it doesn't require to be catalyzed by any enzyme. It just happens. So of course, now here is our 5-fluorouracil, and the 5-fluorouracil will do the same thing. So the 5-fluorouracil will act now and block the imidylate synthetase the same way we have learned. The other way that this can happen is through phosphorylation by the pyrimidine nucleoside, phosphorylation, and that is what is happening is that you phosphorylate this position here. You add a phosphate here. Once you add a phosphate there, that carbon will have 5 bonds, and because that can't happen, then these electrons actually move in, and once they move in, this nitrogen will acquire hydrogen from the surrounding cells or the surrounding environment, and that becomes a H, and that again gives us 5-fluorouracil, and this just cleaves, and there it is. There you have it, phospho-tetrahydrofuran. So that is, again, these are just the ways, just showing us how Tegafar, which indeed is a prodrug, can be converted into 5-fluorouracil. And again, we are reminded, once it is converted into 5-fluorouracil, again, the 5-fluorouracil is active, the necessary enzyme to be activated either to the deoxy UMP, all activated to the deoxy UTP, all, of course, deoxy. And remember, it has a fluorine. So even as we say deoxy, remember it's fluorodeoxy because it has fluorine. And because of the factor that it has a fluorine, whether it's converted to the UMP or the TMP, the triphosphate, the TP, both of them will have an inhibitory activity and therefore stop the synthesis of DNA. Another prodrug is doxyfluoridine, and this is again the structure of doxyfluoridine, same phosphorylation as we have seen of Tegafar can happen. So that is doxyfluoridine, and if you look at the doxyfluoridine, the upper area, the section of the pyrimidine is actually the same. It is only the ribose sugar. It's the ribose sugar which actually is almost a normal ribose sugar. It's almost a normal ribose sugar apart from this position here, because a normal ribose sugar here will have CH2OH. So here is where you have a group. In other words, you have removed the hydroxyl group and you are left with the methyl group, but the other positions are the same. The imidine phosphorylase, the one we have used previously for Tegafar, and bringing a cleave. Once you phosphorate 4 carbon atoms there, you have 5, and this moves here, and that acquires a hydrogen, and therefore you have released again your 5-FU. So the levels of the hydrolytic enzyme thymidine phosphorylase are significantly higher in several solid tumors, such as colorectal, breast, and kidney cancers compared to normal cells. We are only saying this enzyme, there's a lot of it. The levels of this enzyme increase. So it was thought because the levels of this enzyme are high, then you can synthesize a drug, or you can design a drug that can be acted by these high levels of this enzyme. And remember, this enzyme is within the tumors, not within the normal cells. So they were trying to do selective this drug such that once they synthesize this drug, it is acted by the high level of the thymidine phosphorylase to release your 5-FU. And because, of course, the drug is already active, it was, of course, better than the 5-FU itself. High levels of the TP resulted in development. This TP is a thymidine phosphorylase of doxyfluoridine as a 5-FU prodrug. This compound shows GIT toxicity after oral administration due to release of 5-FU by intestinal pyrimidine nucleoside phosphorylase. So the only disadvantage is that you can also get this enzyme within the intestine. And once you get it within the intestines, then you will get your 5-FU already released in released in the GIT, and the 5-FU in the GIT caused a lot of toxicity, including diarrhea. So that was one of the feedbacks or the negative effects of the doxyfluoridine, and therefore not very popular, not not a very popular drug. That was the main drawback. Then you have another one, which is also a prodrug, which is capecitabine. And we look at capecitabine. This capecitabine, again, you can see it shares the same structure as the doxyurea structure here, but it's a cytosine. It's a cytosine derivative. It is a cytosine derivative whereby you can see your nitrogen is derivatized and we can say this is this is 1, 2, 3, 4, 5 pentox. So this is when you put an O, is a pentoxy, but then you add the pentoxy, the last two XL, you put allele. But anyway, let's just say it's an acyl form. Let's make it easier for ourselves. It's an acyl derivative of pentoxy. It's an acyl derivative. That is C double bond of pentoxy. Pentoxy means the pentyl group with an oxygen. So it's an acyl derivative of pentoxy. So that has been put there. And because this is an ester, can even say it's an ester derivative of pentane. And because of this ester group, we know esters are highly susceptible to hydrolysis. So, and you can see, in fact, that is an ester. In fact, it is actually an amide because on this other side we have a nitrogen. So this is that bond is readily cleaved. That bond is readily cleaved, and once that is cleaved, then again you release your cytosine there. You release your cytosine there, and of course your sugar there. So the first activity is by carboxyesterase, and that's when we're saying this is an acyl group and an ester. Carboxyesterase is an amide ester group. And when that happens, then you get the cytosine derivative. And again, the cytosine derivative undergoes deamination. So second stage is deamination, and once by cytidine, deaminase in the liver and in the tumors. So let us note that the first enzyme, the carboxyesterase, is in the liver. But this second enzyme, it's more concentrated. The cytidine deaminase is more concentrated in the tumors. That helps us for a bit of selective toxicity. Because remember, our main problem with these drugs is that the tumor cells and the normal cells very, very alike. So if you do not utilize any slight difference, like any advantage that the tumor cells have, then the drug will have a lot of toxicity. That's why here we are saying the tumor cells have more of the deaminase, deaminase within the tumor cells, and the concentration of this derivative will be much higher in the tumor cells. And then again, within the tumor cells, you have the uridine or thymidine phosphorylase, either uridine or thymidine phosphorylase, and that again now cleaves that to give us our 5-fluorouracil. So there is advantage of the two last steps in that those two last steps allow for a bit of selective toxicity because the enzymes are actually more concentrated in the tumor cells than in the liver, and hence the drug will, the concentration of the 5-fluorouracil will be much higher in the tumor cells than in the normal cells. So it is a prodrug. Again, efforts to circumvent the problems of gastric toxicity led to development of capecitabine. So again, this, as we have said, it is very close to the doxy. You look at this, very, very similar here. You look at it here, can see, as I had already pointed out, very, very similar. So for us to stop the GIT toxicity caused by doxyfluoridine, then they developed the cabecitabine so that we can again reduce of doxyuridine. So that's, sorry, we didn't finish that. So we are saying activated to its cytotoxic moiety, fluorouracil, bithymidine phosphorylase. We already looked at that. It has a high lipophilicity associated with the presence of the this was a group we were looking for, pentyloxy. So I was getting top, actually, they're also calling it pentioxycarbonyl. So it's pentioxy, it's an allyl group, pentioxycarbonyl chain, and is rapidly absorbed and altered after oral administration. So because of this group there, you can see that group again. If you go back, the advantage of that group, advantage of this group, why we are saying there is high lipid solubility, is that portion there. That portion, the pentyl group, so we are calling it pentyl oxycarbonyl group. That pentyl group is high lipid solubility, so it is well absorbed on oral administration. So that is important to note. And is rapidly absorbed and altered after oral administration and metabolized, as we have seen, by the carboxyl esterases in the liver to the 5-deoxy-5-fluorocytidine. And then after that, we have seen it's further metabolized to give us the 5-fluorouracil. Fluorouracil is further metabolized to 2 active metabolites within a normal. Is really not the active drug. It must, as we have said, be converted to the fluorodeoxy-UMP and the fluoro-UTP. And these are the ones which are either taken up, like the fluoro-UTP will be taken by RNA in case the viruses have RNA. They can take up that. And of course, the fluoro, sorry, these are anticancer. So the fluoro-UTP will be taken up by RNA, whereas if you have the fluoro, will be taken up by the UMP. Remember, we also do have in these normal cells, we have both RNA, like the transfer RNAs. We have both RNAs and the DNAs. So for the synthesis of the RNA, you require UMP. It has been fluorinated. It is an antimetabolite, and the same case applies to the deoxy-UMP. It is an antimetabolite. It will stop the process. It will inhibit the respective enzyme, both RNA and DNA, resulting in death of the tumor cells. And of course, some of the normal cells will die. But we have said that because of the advantage of this drug being more concentrated in the tumor cells, and of course, what is called a bit of selective toxicity. But please note that still some of the tumor cells, some of the normal cells will also die. Another prodrug we have looked at another prodrug is the denone 5-fluoro. Again, you can see the fluoro is very important, and it replaces the hydrogen, which is usually at position and number 2, places that hydrogen, which is usually at that position. So then you have your so here normally here you'd have either an amino group or a ketone group. So in there, and then what's of course important is that group at that position. So
Daktari?
Yes.
Casa.
Ooh, sorry. But okay. So what I'm saying is this, we are calling it the 5-fluoro-2-pyrimidinone. We just want to note the differences between this drug here and the normal pyrimidines, or even the difference between that and the kawaida 5-FU, and our position there, it is missing in this group. See, when you make so this is missing in that group there. Of course, we are trying to make a drug that can be orally active, and definitely if you remove the oxygen, of course, you're making the drug to be a little bit more lipid soluble. The drug will be lipid soluble, absorbed, get to into the cells. It is activated into the 5-fluorouracil. So there is, again, you can see the similarities. There are structural similarities and a bit of structural difference. And hepatic aldehyde oxidase after oral or intravenous administration, and you get our 5-FU. Again, we are trying to give it orally for ease of administration. The oral route, as we have said, is the most convenient route that I believe we have done in pharmacology. So that is our 5-fluoro 2-pyrimidinone, and that is converted to our final drug. So we are starting 1, 2, 3, 4, 5. Remember, we're starting to count from that nitrogen there, position 1, position 2, position 3. Now we are starting from 5-fluorouracil. Oh, sorry, the other way around, sorry. Because of the subsequence. Remember, we want the subsequence to have the lowest number. If we go this way, this fluorine you have 1, 2, 3. If we go this way, this oxygen will have 2. So it's position 1, position 2. So the 2-pyrimidinone there, 2, 3, 4, 5. So it's 2,5-fluoro, 2-pyrimidinone, 2-pyrimidinone. So that is the structure there, and converted again fluorouracil, and the story is the same because they are prodrugs. Another drug which is now, this one is we are going back. Remember, we are still talking also of the pyrimidine analogs. We do have another drug which is also a pyrimidine analog, a cytarabine cytosine, cytarabine or arabinosyl cytosine. So you can either call it cytarabine or you can call it arabinosyl cytosine, and the arabinosyl cytosine is the one which gives it this short name, RC, arabinosyl cytosine, gives it that short form of name. And it is a pyrimidine again analog, and it is a cytosine arabinoside. It has a very slight difference by the arabinose sugar. You have the arabinose sugar in place of ribose sugar. And the difference between arabinose and ribose is that the arabinose is an epimer. Remember when we talked about epimer? So arabinose at position number 2, where you have a hydrogen and a hydroxyl group, the hydroxyl group, there is epimerization of the hydroxyl group such that, and that changes the ribose sugar to arabinose. So the only difference in structure is an epimeric. So that the 2-hydroxyl group has undergone epimerization to give us the cytarabine, and that gives a difference. Once you change the epimerization, or once you change the orientation of the 2-hydroxyl group, that change of orientation changes the way that drug binds, and therefore changes it completely. And indeed, it becomes an antimetabolite and not the proper metabolite. This epimeric sugar is similar enough to the natural ribose to allow arabinosyl cytosine to be incorporated into DNA. So, of course, once it is incorporated, again, we are saying the same thing. Once it is incorporated, it is not the right metabolite, and therefore it will bring a termination of the DNA synthesis. So there will be slowing of the DNA chain elongation reaction via DNA polymerase, and therefore there will also be cellular inefficiencies in DNA processing or repair after incorporation. So those are the same thing will happen. What is important is to look to understand the various drugs, the structures, and how, of course, they are bringing about the same change. So these are cytosine arabinoside. So this here you have position 1. Who is there? Just 1 minute, students.